Today I want to talk about the immune response to helminth infections, so to infections with worms, and in particular to helminths that lead to extraintestinal infections. So not those ones that are simply in the gut, but to the ones that actually go to the tissues. And you can quickly think back to microbiology. What would be examples for such helminths? Okay, so what might come to mind is Toxocara, Toxocara cani or cati. So those worms that children might pick up if they play on playgrounds where cats and dogs are, well, doing other things than playing. Or if you think about the tropics, schistosoma is an important one. So this helminth that can infect you right through the skin. Okay, before we start, question to you, and you can pause the video and think about this. What is the most important type of leukocyte against helminths? And so important, I guess you can always argue a bit. So what I mean, what is the leukocyte that mainly directly fights helminths? Okay, the answer that I'm looking for is, of course, eosinophils, eosinophil granulocytes. In my perspective, is one of the subjects that you have to learn in layers of complication. So you start with the basic principles, then you add something on top, then you maybe learn about some exceptions to those principles that you learned early on. It doesn't make so much sense to try to learn every detail just in one go. That's just too much. And that's also the spirit that we're going to take for this class. So we're going to simplify this subject of fighting the helminths by saying the eosinophils are the most important leukocyte, the most important directly acting leukocyte, and everything else that we look at is how do we recruit and how do we activate these eosinophils. And all the other details, all the other mechanisms that are there, we're kind of just going to ignore, but that's going to cover quite a lot of ground, even so we're not going to cover everything. So let's just assume we have our worms here, and these worms are in the tissue. And I should already say here, everything that I'm drawing now is not going to be to scale. So if this was to scale, the worms should be much bigger. But okay, so we have these worms in tissue, and somewhere nearby there is blood vessels, there are capillaries, and in these capillaries we have our eosinophils or our eosinophil granulocytes, and as you might know, for granulocytes, these always have this bit weird looking nuclei. So what I'm trying to do here is to represent this nuclei kind of in the form of a G, so granulocyte, and then we're going to draw in here granules, because that obviously they're granulocytes, so they have granules. So what could be the first mechanism that we can recruit more of these eosinophils into the tissue? Well, a first step could be to simply make those blood vessels, these capillaries, bigger. So we want to have a swelling and dilation of these capillaries. So let's take off the previous one. Okay, so now we have our eosinophil in bigger capillaries, just so that more eosinophils can come to this tissue. Next step, and I'll just draw these as different, so these different mechanisms as different sections of capillaries. The next step would be that we're expressing adhesion molecules on the endothelial cells of these capillaries. So this could be selectins, this can be VCAMs, so vascular endothelial adhesion molecules, and then our eosinophil is going to get stuck here. And in reality, so it means these eosinophils can still move in the direction of the blood. So typically they have this rolling motion along these endothelial cells, but it's slowed down. So they're moving much more slowly than if they were just flowing by with the speed of the blood. Okay, Next mechanism would be, now we also want to have these eosinophils to come out of the capillaries and go into the tissue. And this is achieved by contraction of these endothelial cells. And through these contractions, we'll have opening of gaps between the endothelial cells. And what can happen now is that our eosinophil can squeeze through here. So it can transmigrate out of these capillaries and go into the tissue. So again, these three mechanisms. The first one would be dilation or vasodilation. The second one is the expression of adhesion molecules. And the third one would be an increase in permeability or vascular permeability. And then this diapedesis or this transmigration. Okay, eventually these eosinophils will then come close to our helminths. And what is happening now is that they're degranulating, so they're emitting the substances that have been in these granules to then kill those worms. I asked you, why are these eosinophils especially suitable for killing worms? Well, that's because they have substances that can attack these worms. They have several substances that we also find in other granulocytes, so those typical enzymes, these proteases and so on, that can fight whatever is in their surrounding. But in particular, they have what we don't find in other granulocytes, the eosinophil cationic protein and the major basic protein. And those ones are specifically suitable to fight helminths, to destroy the tegumen of helminths. These proteins, so we have one protein that has cationic in its name, one that has basic in its name, so these are positively charged proteins, and that is the reason why those eosinophils are eosinophil. So they are binding to eosin, so which would be at physiological pH negatively charged, so it binds to this cationic or basic proteins, and that's why eosinophils are stained with eosin. Okay, good. So now we have understood several of those mechanisms that are going on here. Now, what causes these mechanisms? And the central role in triggering these mechanisms comes from mast cells. So mast cells, they're also cells that carry a lot of granules. They're not granulocytes, but also carry a lot of granules. And they are cells that are typically found in tissues, not so much in the bloodstream. And so these mast cells are responsible for the signaling mechanisms. So they are signaling on the one hand side to the endothelial cells for this dilation. So what's actually happening is they trigger the endothelial cells so that these endothelial cells themselves signal to the surrounding smooth muscle so that the smooth muscle relaxes, and then the effect is this dilation. They signal to the endothelial cells to express these selectins and VCAMs, and they signal to the endothelial cells so that they contract, so that it comes to this increased permeability. Increased permeability, this of course means not only that now cells can come out more easily, but also that liquid comes out more easily. And what's happening then, there is swelling, or we can also talk about an edema, which is often one of the complications of inflammation. So this increased permeability, they're very swelling, and if you have that's leading away now from the helminth infections, but if you have a disease where you have like over your whole body many sites where you have this inflammation, then you lose a lot of liquid into your tissues, blood pressure drops. But okay, that's not so relevant for right now. What else do these mast cells do? So we said this signal to these endothelial cells. They actually also signal here to those granulocytes to attract them, to lead to chemotaxis. So on the one hand side, that these granulocytes come out of the vessels and into the tissue, and then usually these mast cells would be close to the worms, so they even attract them down or up a chemokine gradient to come close to the worms. I don't want to attempt to tell you, okay, this signaling molecule is more important for this process and this for the other process, but in general we want to have a quick look at which signaling proteins, or which signaling factors are there. And probably the most well-known one as a mast cell released proteins on one of these mast cell signaling factors, one that's already preformed in those granules, is histamine. Histamine molecule typically associated with swelling and vasodilation. Then we have lipid signaling molecules, and there we have prostaglandins. So in particular, prostaglandin D2 is an important one here. And prostaglandins, so the synthesis of prostaglandin, there is a famous drug that inhibits this synthesis, and this drug is called aspirin. So aspirin is involved in inhibiting the formation of prostaglandins from arachidonic acid. So arachidonic acid, a lipidic acid. So now you might ask, what's headache got to do with it? And the answer is everything, kind of, because headache, the basic underlying cause for headache is also this swelling that we just talked about in context of the permeability. So aspirin inhibits the formation of these prostaglandins and thereby inhibits swelling. And swelling, as I said, the underlying cause also for headache. Okay, plus prostaglandins, then important also leukotrienes. There are also different ones of those, also a lipid signaling factor. And then we have some other ones. One more important one, especially for this chemotaxis, is PAF, so the platelet activating factor. These are the important ones of these mast cell signaling factors. In addition to mast cells, what else do we have? Which other cells that are relevant for this signaling? We have here Th2 cells, so T helper 2 cells, and also ILC2, so innate lymphoid cells too. So before we continue with the Th2 cells, here's a question to you for some recap. What are the signature cytokines of Th2 cells, of this class of T helper cells? And then try to remember briefly what are those functions of those cytokines. So just take 5 to 10 minutes to think about that. Okay. So what are the signature cytokines? The signature cytokines are IL-4, IL-5, and IL-13. What are their functions? And thinking about those functions, I want you to essentially think of this figure 10.9 in the current edition of the Abbas, which is, my opinion, the most beautiful figures in this book, and also this chapter about the different T helper subsets, this chapter 10, is a really good one. Okay, so you might think back, okay, there is this function of IL-4, IL-13 mainly in triggering peristalsis and mucus productions. And this can also, be very relevant in the context of Helminth infections. In this case, it would be more those Helminths which are intestinal rather than extraintestinal, what we're talking about here. Then there is another one, which is this alternative macrophage activation. So this activation of macrophages, this M2 macrophages, which are more associated with repair. Okay, there it might not be directly clear how does this have to do with the Helminth infections, but actually it can have. So by this production of the extracellular matrix of these M2 macrophages, they actually can kind of form a layer around these Helminths, which we're not talking much more about here, but they can form this layer and kind of lead to granulomas. So they can contain at least these worms, because worms are often very difficult to eliminate, despite of what we're talking about here with the eosinophiles. So this is actually also a mechanism, not just repair, but also containing pathogens, which is relevant in this Th2 signaling. Okay, then we have various functions of IL-5, which we're going to talk about next, which directly has to do with the eosinophiles again. And then there is also important functions of maybe not so much Th2 cells, but TFH cells, so cells in the lymph nodes, which are producing these cytokines, so IL-4 and IL-13 mainly, and which play an important role in this class switching to IgE, which I'm also going to still mention a bit here. So let's talk about these Th2 cells and, as I said, similar functions from ILC2 cells. ILC2 are believed to be more tissue resident even than the Th2 cells. So one function which is still a bit debated, still not quite that clear, is involvement of the Th2 cells also here in the expression of those adhesion molecules. So this may probably be a function of IL-4. And then the much more established functions are the activation of the eosinophiles, so activating them towards this degranulation, and this function is mainly through IL-5. And another important function of IL-5 is the stimulation of the production of those eosinophiles. So if we draw our bone here, and of course again clearly not to scale, oops, and a bit bulgy. So here also we have IL-5, and then we have increased production of eosinophiles in the bone marrow, so that we just end up with more of these cells here in the bloodstream in the first case. And so, speaking about IL-5, think about the beehive, so the E in the beehive, and then you can think about, okay, functions of IL-5 eosinophil production. If you have no idea what I'm talking about, I'll put you the link to this video by H.A. Monix, which is one of my all-time favorite YouTube videos, and great in helping you to remember a number of important cytokines. Go one step even further back, and let's ask, okay, those cells, so those mast cells, those Th2 cells, how did they get activated towards the signaling that they're performing here? And for this, it might make sense to perform a division. And on the one hand side, we're looking at the tissue that we were looking at before, so with our worms. And on the other hand side, we'll look at the lymph node, which I'm going to just represent as something like that. Okay, what we have here in our tissue where we have the worms, we have early on, let's say, dendritic cells, so they have these spines, and those dendritic cells encounter worm antigens, and at the same time they get stimulated by epithelium generally. So this one could, for example, be the lining of the gut, and let's imagine we have some more of our worms here in the gut, for example, and then this epithelium is going to signal to those dendritic cells, and the main cytokines here are 25 IL-33, and it's believed that these cytokines kind of push this dendritic cell in a certain direction, to promote the Th2 response. So what's going to happen is those dendritic cells go into the lymph node, and these dendritic cells that have been stimulated in this way are now in the lymph node, stimulating on the one hand side Th2 cells, and on the other hand side, TFH, they're stimulating TFH cells in this direction of signaling in this in this mechanism that involves these Th2 signature cytokines, so in particular IL-4 and IL-13. And using these cytokines, so as I said, in particular IL-4 here, they are now signaling to B cells, and this signaling now results in B cell class switching to the production of IgE. So this is again one of the things where you can think about HA monics, IL-4, DOR, beg for help, so stimulation of B cells towards the production of IgE. These B cells will now leave the lymph node again and go to the tissue, convert to plasma cells once they also encounter these warm antigens, and they are now producing these IgEs, and these IgEs are bound by plasma cells, so they have these Fc receptors on their surface and they bind IgE. And now if we have the binding of, again, these antigens, then this leads to stimulation of these plasma cells. Strictly speaking, what has to happen here is that they are cross-linked. So we have cross-linking of IgE antibodies, which, in this case typically recognize the same type of epitope, so that brings them together, and then this one will lead to the degranulation of these mast cells, these factors that we talked about before, so the histamines, and at the same time also the production and release of these lipid mediators, for example, so the prostaglandins, the leukotrienes. In this context of Helminth infection, we also have to think about, okay, there are also other mechanisms of activation of mast cells, so there is actually a number of activation mechanisms. This IgE is the most typical and most well-known, but there can also be activation through complement proteins, for example. There can even be activation through neuropeptides, so something like somatostatin, which kind of provides an interesting link between, well, neurology and immunology. Yeah, so the other thing that we said is, okay, back in the lymph node, we had the stimulation of these TFH cells, but also Th2 cells, and then the Th2 cells, they are also going back to the infected tissue. They are going to there encounter those dendritic cells as well, which are doing the antigen presentation, and the Th2 cells are going to recognize that and are going to be activated in this way. Ready to recap what we talked about. So the first point was the eosinophils. The eosinophil granulocytes are the main leukocyte that directly attacks these worms, these helminths. Then we said important mechanisms in getting those eosinophils there and having them attack these helminths is the dilation, so the widening of the blood vessels, the expression of cell adhesion molecules on these endothelial cells of the blood vessels, the increase in vascular permeability, then the stimulation of the degranulation of the eosinophils, and the stimulation of eosinophil production back in the bone marrow. We said the major cells that trigger these mechanisms are, on the one hand side, the mast cells, and on the other hand side, those Th2 cells that we can find in tissue. For mast cells, we said they mainly mediate these mechanisms through histamine, which is already preformed in their granules, and through lipid mediators, especially here the prostaglandins and the leukotrienes. And then we also have additional cytokines, and particularly important here was this PAF, the platelet activating factor. And for the Th2 cells, we said, okay, signature cytokines, IL-4, IL-5, IL-13, and in particular here was special importance was placed on the IL-5 for triggering the degranulation of the eosinophils and also stimulating the production of more eosinophils.